afternoon, everybody. Well, before I start, okay, let me see if I can figure this out. So this is. First of all, I would like to thank, actually, Joe for inviting me here. It's a pleasure and an honor to be here. I mean, it's a fantastic conference. I learned so much this morning. And actually, this morning was a very nice introduction and to some of the things that I'll talk to you about this afternoon. Also, I want to thank Bill Polvino for introducing me to Joe, I don't know, like a few months ago. Of course, I had heard of Joe and the great work that he does. But it's really good and really fantastic for us to have this interaction with the Cyber Center. Because as Joe was mentioning, Takeda has an emerging interest in neurodevelopmental disorders. And I'll tell you a little bit about it. I'll tell you a little bit about the way we think about it. It's probably a little bit different than what other companies have done. But I think that's where we need to start moving towards too. Oh. Oh, the green one, of course. All right, so the title of my talk is Patient-Focused Approaches to Drug Discovery in Neurodevelopmental Disorders. I think the talk is like around 20 minutes, 25, and I want to leave some time for questions, because I heard a lot of questions this morning about therapies and availability of clinical trials on neurodevelopmental disorders, and I'm happy to answer whatever questions you guys have on those topics if I can answer them. So the outline of my talk is here. So I want to give kind of a landscape, and I'm sure I'm missing some of the activities that have happened over the last few years, but kind of like a high-level overview of the clinical landscape in autism and in neurodevelopmental disorders. And by doing that, I hope you will appreciate changes in the level of investment over the years. Then I'll put a list of challenges in neuroscience drug discovery. So drug discovery is very challenging, as Bill explained this morning. But drug discovery in neuroscience, it's even more challenging. But we think there are certain advantages of using genetic syndromes, and actually neurodevelopmental disorders can be a great platform to start building successes. Then I'll kind of reiterate Joe's message this morning that genetics are actually key to understand the biology and that we need to take advantage of those. And hopefully you'll see how we kind of do that, how we take advantage of genetics. And that kind of leads us into the use of tools like patient-derived cells, patient-derived systems to increase the probability of success in the drug discovery process. So this slide here, actually Rob Ring, who was Autism Speak CSO, and I made together, I don't know, like last year or so. And it kind of tries to illustrate changes in investment, which is kind of like the shaded area here, together with what has been going on in the area of autism over the last, I don't know, 20 plus years. And as I said, I'm sure I'm missing some, so I apologize for that. But there is a point to make in this slide, which is this valley here. And obviously, this valley here represents what happened after the Fragile X trials were not successful, both at Roche and Novartis. And it kind of exemplifies the fluidity that exists in pharmaceutical industry in terms of moving funds from one indication to the other, depending on where we are successful and where we are not successful. So, and that brings me to this concept of if we are successful, more money is going to flow in. If we make mistakes when it comes down to choosing targets or choosing biomarkers or whatever it's involved in success of a clinical trial, that's going to basically scare people away from indications that are not successful, and that's just the reality, the sad reality of the business. But then the other aspect important to make in this slide is there's been actually a very strong uptick in the last 5 years in the investment in the industry in these disorders, and exemplified by a few of these trials that are explained here. Now, what are companies going after in terms of indications, or the way we kind of split, or the way I split indications or focuses in the neurodevelopmental space when it comes to the clinical pipeline. So these are two tables that basically divide trials ongoing, and again, I'm sure I'm missing some, that are focused on idiopathic forms of autism. And then here is a list of some trials that are ongoing in genetic syndromes. And this is pretty common. I mean, this is the way the industry has functioned over the last few years. And there are advantages and disadvantages in each one of those. Obviously, going after the idiopathic population, you have a large population, so it's a market consideration. Challenges in patient recruitment, maybe less, you may have less challenges. There's some endpoints that are somewhat validated, and this is an area that clearly this autism and developmental disorders needs to work very hard from now on. There is some regulatory precedent with risperidone approved for rejectability, and breakthrough designation is still available. However, if you focus on the genetic syndromes, actually, I think there are certain advantages in this area as well. Obviously, there is regulatory incentiveness. There is a lot of work being done by patient organizations in terms of getting people trial ready. And then the targets are informed by human genetics. And this is where Takeda, at least the group that I lead, focuses on. We're trying to generate therapies starting on genetic syndromes. And the main reason actually is because that's where the biology is leading us. That's where the data is leading us. You may ask, well, none of these targets actually go after the genetic mutation or anything related to it, or not directly related to it. And I think that's the change that we need to make now, which is come up with therapies for genetic syndromes with therapeutic agents that actually go after what the mutation is driving, or whatever is the downstream consequence of that mutation. Okay, so having said that, and I'm also an optimist, Bill, but there are a lot of challenges, as I mentioned, in CNS development, drug development. And I'm trying to exemplify here 5 of them. So the first and most closely to what I work on and I have more expertise on is poor disease biology understanding. What do we know about schizophrenia? I mean, we know a few things. We know genetics are important, but we don't really know. There's probably like many disorders within schizophrenia or Alzheimer's disease, right? You all have seen all the amyloid therapies failing over the last 20 years. Probably we don't know exactly what those disorders are about. Heterogeneous patient populations, as I mentioned, many of patients that are in many disorders, we have patients that probably have different types of disorders, and that creates a lot of noise in the clinical trials. Clinical endpoints, precision of the clinical endpoints is low. Many times the clinical endpoint are questionnaires to our patients that in many cases it's difficult for them even to communicate. So that's another area that it's common in CNS drug discovery. Operational challenges. The trials, many times it's very difficult to run these trials. I was once in a meeting from one of our colleagues in clinical trial, and he was explaining us how the same patient was enrolled as a placebo, or was enrolled twice, and that person was receiving placebo and also active drug. And so that's going to create a lot of noise too. And so we need to be much more efficient in operating the clinical trials. And then very importantly, actually, is also many times the payer doesn't think that the outcome that we are providing is relevant. And so thereby, that payer is not going to reimburse. And then obviously, companies will have a tough time, making what they do, which is making money. So the point, I guess, I'm trying to make is actually that neurodevelopmental disorders and genetic disorders solve some of these problems. For example, and as has been mentioned this morning, much better than I can, Joe and Stormy have shown you how in these disorders, we have a very direct link between genetics and disorder. And we know what's happening in the patients. We know what originates, we know what originates the disorder. Obviously, patient stratification is a little bit simpler because if we can genetic test the patients, then we know what kind of mutation they have. So that's a good way to segment patients. There is a hope and some work ongoing that talks about or is trying to understand patient-specific biomarkers. Can we identify a biomarker? Can we identify something that we can measure in those patients that our drug can modulate that is specific for that population? Patient organizations are key in these syndromic forms. So we think that operational challenges in clinical trials is a little bit less. And obviously, there is a favorable payer environment because there is a very important clinical need. And if we come up with a therapy that makes an impact on the patients, the payer will reimburse. But rare disease drug development actually requires big investment from companies. And so that's something that all of us need to take into consideration. There is a big effort in patient engagement, as I mentioned, discovering biomarkers, understanding outcome measures, how the patient changes over their life, what kind of symptoms do appear as the patient evolves and grows up. Those many times are not very well understood. So that requires investment from both patient organizations, from public groups, as well as companies. Preclinical models. Many times, actually in this area, preclinical models are a little bit more relevant because, as I mentioned, you can recreate the genetic, at least some of the background, not the common variation, but at least the mutation that drives the disorder. You can recreate that in preclinical species, and you can study what happens to those models. Prevalence, that's actually it's an area that I'm not an epidemiologist, but I'm always surprised by the fact that we don't know many times how prevalent these disorders are, and that having an accurate estimate, it really helps to move these programs forward. And then clinical operations is challenging, right? Many of the big pharma are used to big disorders where it's relatively simple to recruit. In this case, you need a much more specific and probably involved team. So these are just things that, when we think about building programs and building, so somebody was asking this morning, like, are companies developing drugs for neurodevelopmental disorders? Yes, there are companies out there doing it, and very well. But it requires big investments to be successful. So how do we approach it? And I don't know how, because I've been in the industry for 8 years now. I work for a company Lundbeck, which we were in New Jersey, and actually we were working on neuroinflammation. Then I moved to Takeda 6 years ago, and I don't know a lot about how other companies approach these disorders, but I know how we do it. And so we start with genetics. So we want to focus. We want to focus on disorders that have a genetics that are driving the phenotypes. And why do we do that? It's easier just from a selfish standpoint, from a scientific standpoint. It's easier for me to conceptualize what I'm studying. If I don't know what is driving the phenotype of my patient, how do I start? I don't know how to start. Here, at least I have that basis, which is that's important for me. So that's how we start. Then we do human-centric drug discovery. So that means we use human cells or human systems, and I'll show you a couple of examples of those, to do drug discovery. We can go after a single target, or we can think about phenotypes. What is different between the cell of a patient versus a cell from a typical developing kid? And understand how that difference occurs and how we can modulate that. We think that can create potential new lead in therapeutics. As I mentioned, that basically helps us into stratify patients using genetics, so that's a problem that gets more or less solved. And it leads us into the idea that then in these patients, we'll have a biomarker, which a biomarker is something that you can measure in a patient that may or may not be related to disease progression, but it's manifested in the patient, and that you can modulate that with your compound. So that's very important to move programs forward. Obviously, as I mentioned, increases patient homogenization, because we know that those patients they all have a similar type of genetic background, which hopefully brings us to a little bit better defined endpoints, clinical endpoints that are a little bit more relevant, which helps payers to justify reimbursement. And it leads us into precision medicine, which has come up a couple of times today. So this is kind of where the industry is moving today in neuroscience as well, precision medicine. We don't want to treat millions and millions of patients with the same drug, because we know that that's unlikely to be successful. We want to have therapeutics that are highly effective in homogeneous patient populations. And obviously that is not possible without patient organizations. Actually, patient organizations helps us in both ways. They help us understand genetics. They help us understand what are the relevant symptoms in the patients. And they helped us in many other ways as well that I'll show you later. So that's kind of the virtuous cycle that we are trying to build within Takeda, using these neurodevelopmental disorders as kind of like a testing ground. I'll talk just very briefly around genetics. So this kind of shows how the prevalence of autism has changed over the last years, and this is because of many reasons. Obviously, we are now starting to understand genetic risk architecture, and I'm not going to get into this, but obviously, Joe already talked about concordance rates between twins, and et cetera, which underlie a lot of the genetics in autism. Obviously, strategic resources and initiatives that have happened over the many years have helped to drive funding and to drive research and understanding in this area. And then, again, the revolution in genetics actually has helped us to understand, the genes involved in these disorders, and that, that kind of goes hand in hand with advances in genomic technology. Basically, this has happened a revolution in genomics that has helped us to increase this. So, this is a nice graph that I took. So we built this out of the Safari web page, but it just helps identify that, as Joe was mentioning this morning, we have a lot of single gene syndromes actually that are amenable for a therapeutic if we can really understand how to do it. And the good thing is that many of those, as you very well know, have patient organizations behind them and that support research, support access to patient samples. They help us in access to patient registries, recruitment. They have biorepositories. They help us understand about the outcomes of the patients, understand clinical endpoints, which is very important. They can be partners in research, in the patient journey. Also very important, natural history studies. I mean, this is an area that we need in order to understand the patients. And also they educate regulatory partners. So, we call it force multipliers. This is the way we want to work, and that is one of the reasons that working in these areas, we think it's highly beneficial for everybody. Now, how do we think about clinical targets, or what? Because one can think about, okay, we want to treat autism, but it's unlikely that we're going to treat autism. What we want to treat is symptoms that these patients have, right? And this is, you probably have seen this in the past several times. This is kind of a representation, a graphical representation of a variety of symptoms that are, or clinical targets that one can go after. But at the end of the day, that doesn't matter if you don't improve quality of life, right? And we heard a couple of times today talking about respect and dignity, inclusion, academic success, residential solutions. So at the end of the day, there needs to be a connection between improving any of these symptoms and having a better life for our patients. And that may be different for different syndromes, right? So some syndromes here, put an example of Phelan-McDermid. Some of these syndromes are more, they have a stronger effects on, pain or seizures or hypotonia, and others have, like in Rett syndrome, you have changes in respiratory behavior. So we need to understand how these different syndromes differ, and that then help us bring the bridge from preclinical to clinical. Obviously, we all hear about that this autism is a very heterogeneous population, and it is. But the way we see it is that it's basically a spectrum of rare psychiatry and rare neurology. And we think that they may have different features, but actually, in many cases, they may have a common underlying biology, and that we can go after those biologies to come up with novel therapies. Now, translation is a challenge. So I work in the more early parts. I'm trying to understand a little bit the targets, but you need to also understand that one needs to translate whatever happens at the preclinical space into the clinic. And that is an area that there is a lot of active efforts. And there are several opportunities. There is work on 3D cell systems and understanding electrophysiological properties and EEG properties and other biomarkers in these systems. We need to understand things like inflammatory status of animal models, how that correlates with patients. How do we image if we change things like synapses or if we change neuronal activity? There is a lot of work to be done in that aspect. How do we approach this? So we basically use, and Stormy gave a very nice introduction to this concept of patient-derived cells. So if you work in oncology, you can take a sample from a tumor and understand what the tumor does. If you work in dermatology, you can take a sample of skin. If you work in a brain disorder, you cannot take a sample of the brain because for obvious reasons. But what we can do nowadays is actually reprogram cells either fibroblasts or either blood cells into neurons or other cells of the brain and understand how those behave. And we can actually even do circuitry, and I'll show you an example of that. So these are the type of experiments that we do. I don't know if this shows very well, but so I don't know if you guys can lower a little bit the lights. But yeah, very good. So basically what it shows here in red, you have neurons, and then in green, we are imaging synapses or spines or protrusions, probably protrusions. And so we can ask questions how our therapy changes those protrusions, how the mutations change the protrusions, and study what compounds would do in that area. So these are 2 videos. I don't know if we can play them. I don't know if I can play them from here, probably not. But that show a 3D system. So these are 3D, these are balls of cells that basically oscillate. And one is from a typical developing individual, and another one is a mutation specific. And don't pay attention to the size in this case, because I think the images are taken at a different time of modulation. But we can study these types of, and these are from human cells, right? So these are what people call mini brains, and we can study these samples. And we can study them from, mutations. We can put compounds. We can do all sorts of modification here to understand the biology. Oh, there you go. Oh, so that's what I had to do. Press play. So, so that's how these things look like. Okay. So you have these, these oscillations, and you can measure those in the lab and do all sorts of studies. So I guess take-home messages, I see like a 000 there. CNS drug discovery is very challenging, but actually advances in genetics and in novel therapeutic modalities are open the door for new medicines. And I think that's probably the reason that many people in the industry are actually getting very interested in these areas. I think the use of patient-derived cells is actually generating disease-relevant knowledge that will be key for novel therapeutic discovery. That basically means that in the past, we have been generating a lot of rodent-relevant knowledge, which has led to not so successful clinical studies. And the idea is that by using human knowledge, will increase our probability of success. I'm not saying that we will be successful, but it probably puts us in a better place. On the other hand, translation and clinical work is still challenging, tremendous challenge in this area. Joe touched briefly on time of intervention, and yes, we agree that these patients, the brain is plastic and you can change, but I think obviously time of intervention is still a challenge. And also availability of clinically validated biomarkers, they do not exist nowadays still. And so working towards that is going to make a big difference for us. Now I'll stop here and I'll take a few questions.